Now, when we talk about culture media, when you need to grow a bacteria inside a lab, you sort of want to give it nutrients that are suitable for that bacterium or a group of bacteria. And for that, you require this culture media. So it is basically a source of nutrients, and it will contain all the different kinds of nutrients like macronutrients, micronutrients, and all the supplements that are required. So it can be in the form of liquid media, which are broth, and it can also be in the form of solid media or semi-solid media. So we use agar, which sort of solidifies the liquid media into a solid media. Now, these media, they can be classified according to a lot of things. First, let's talk about the classification based on chemical composition. Chemical composition. Now, according to the classification of chemical composition, the media, they can be defined or complex media. So the first one is defined media, and the second one is complex media. So in case of defined media, what happens? So for defined media, in these media, the ingredients that are used, the ingredients, whatever it is, these ingredients would be of a known or defined chemical formula. So we know what we are giving to the bacteria. We know the exact defined chemical formula. So for example, let's say we are growing some photosynthetic microbes that uses carbon dioxide as carbon source and light as energy. So we're going to give them sodium bicarbonate, the formula we know. We give them nitrate or ammonia as nitrogen sources. We give them sulfate. So all of these we mix together and we make the media, and that is why it is called defined media, because the composition of the media is known. And this is also called, since we are making the media, I mean, we are literally giving every bit of salt, every bit of supplements, and making the media. So these kind of media are also called synthetic media. So that's defined media. Now let's move on to the complex media. Now, in contrast, complex media would have some ingredients known while others would not be known. I mean, it has got some unknown chemical composition. So, some ingredients undefined chemical composition. So, for example, if you have worked in a microbiology lab, you would come across something called beef extract, or yeast extract. So, these are extracts, and you do not know the exact chemical formula or what is there in the extract. You don't know about that. So, I mean, you don't know the precise chemical formula for that. So that is why these are called complex media. And you probably would have used media like nutrient agar and nutrient broth. So all of these are complex media. Most of the media that we use in a microbiology lab, most of them are complex media, until and unless we use something like 2% sucrose agar or something similar to that, which is a defined media again. So based on the chemical composition, we have these two, the defined media and the complex media. Now, moving on to the functional type. So moving on to let's make some room. Functional type. So this is the second one, functional type. There can be a lot of types. So the first one is supportive. So in supportive media, these are also called general purpose media. General purpose media. So these media, they can sustain the growth of many types of microorganisms. So that is why they're called general purpose. So they're not specific to a particular microorganism, and that is why they are sort of a general type. And the example would be tryptic soy broth. So tryptic soy broth, this is a supportive media or general purpose media. Even tryptic soy agar. So these are general type media. And there is also something called enriched media. If you've heard about blood agar media, which contains 5% sheep blood. So these media, they sort of provide some extra components which are present in the blood to encourage the growth of some fastidious microorganisms. And we can use this blood agar to look at any bacteria, whether they're causing hemolysis or not, whether it be alpha hemolysis or beta hemolysis. The third type is selective media. Now, selective media, they would allow certain organisms to grow, but they will also kill certain organisms. Say, for example, we have EMB, eosin methylene blue agar, and McConkey agar. So these are, let me write that down, EMB or McConkey. So these media, they sort of they kind of help the gram-negative bacteria to grow, whereby suppressing the growth of the gram-positive. Right. So this eosin methylene blue, they have the eosin Y and the methylene blue, which is kind of toxic to the gram-positive. And McConkey has got some bile salts which are toxic to the gram-positive. So that is why these are selective media. So they are selective towards what they are allowing to grow and what they're killing. And finally we have the differential media. Now again, this EMB or McConkey, sorry, these fall under differential media as well, because they provide some differentiation between different kinds of bacteria, whether they are lactose fermenting, whether they're lactose non-fermenting, and that is how we can distinguish by looking at the plate.